Now moving towards another important topic, which is very important difference between primary and secondary colonizers. So let's start it. Bismillahirrahmanirrahim. So basically, the first bacteria that colonize in the it is basically non-pathogenic. They actually make the conditions favorable for the upcoming bacteria that actually cause the destruction of periodontal tissue. They are the early colonizers attached to a tooth surface and lay the foundation of the growth of biofilm. They actually periodontal pathogens cannot attach to the biofilm or accumulate until these non-pathogens are attached. Means you tell the students to alphabetically line up like it includes the species that cannot survive without oxygen. So this is actually a mistake. Actually, these it includes those species that can survive without oxygen, that are obligate anaerobes. Hemophilus, Neisseria, Actinomyces, and Velanola species. Okay, what are the characteristics? Actually, they provide the new binding sites for the other oral bacteria. They modify the environment by what they actually they modify the local environment and influence the ability of other bacteria to survive in the dental plaque by a flame. For example, what they do? They remove the oxygen, they make the oxygen tension low, so the survival and growth of obligate anaerobes become easy because those bacteria actually cannot live in the presence of oxygen. Gram positive actinomyces and streptococcus and bacteria began to multiply as intermediate colonizers. Gram negative bacteria Physobacterium and Provatella intermedia, and gram negative bacteria then began to colonize is Porphyromonas and Capnocytophaga. Okay, together with the growth of these microorganisms and cohesion, it will lead to the development of microcolonies and a mature. Secondary colonizers are Provatella intermedia, Provatella lozenci, and Capnocytophaga, Fusobacterium, P. gingivalis. They do not initially colonize in cleat tooth surfaces but adhere to the bacteria already in the plaque mass. This is a diagram showing association, adhesion, and proliferation. Then microcolonies formation, then bioflame mature formation, and then growth maturation. Then sequence of attachment within the arch pellicle forms over the crown. Then the early colonizers comes supragingivally, and within the 6 days, it's actually the tooth is covered by a layer. In 7 days, there is a mature supragingival biofilm, and in 3 to 12 weeks, subgingival biofilm starts to form. So, moving to another important topic, that how does the gingival inflammation favors the plaque formation? When there is accumulation of a lot of plaque in the gingival margin, so there is an inflammatory reaction of the body of the soft tissues. In the presence of this inflammation has influence on the local environment and ability also of the blood and clavicle fluid, promote the growth of gram-negative bacteria and increase the periodontopathic potential. The plaque formation is more rapid on the tooth surfaces that is facing the inflamed gingival margins as compared to plaque that faces the healthy gingiva. Studies shows that during inflammation, the clavicle fluid increases during the production that enhance the plaque formation. Probably some substances from the exudate, like you have minerals, proteins, carbo, favor the initial adhesion and the growth of early colonizing bacteria. Conclusion is basically that immediately following immersion of hard and non-shedding surfaces into the fluid, there will be adsorption of micromolecules that will lead to the formation of biofilm. The bacteria adhere to this glycoprotein layer, which is actually a pellicle. First involve the primary plaque formers such as gram-positive fecalated cocci and rods. Okay, then the subsequent colonization leads to the receptors. Onto these receptors of these organisms will involve gram-negative and strictly anaerobic bacteria, while primary also multiply to form colonies. Periodontal pathogens we know Acrogatibacter, Actinomycetes comitans, Porphyromonas gingivalis, and Teranella forsythia.